I'm going to talk about variability between multicellular organisms, which you've heard a bit already this afternoon, but this time in plants. So I'm not expecting you to become plant expert by the end of my talk. So any name of gene you can forget while I'm saying them, except if you want, I'm going to use as another gene. And so before going into the details of what I'm going to explain, just to put a wider context of what we are looking at. So, as I said, we are interested in variability between multicellular organisms, which we can find at the level of the phenotype and at the level of expression. Usually, I'm mainly working at the level of expression because it's easier to understand what's going there. Because looking at phenotypes, it's harder to understand what is controlling each phenotype specifically when it is a phenotype where you have hundreds of genes probably getting that phenotype. And so what we're interested in is to understand what is making this high level of variability in expression between plants, what the factors that are involved in there, if it is also changing in response to the environment, and then if we have a link between this variability in expression and the variability in phenotypes. So obviously that's a lot, and I'm not going to talk about all of that today, just three main points. The first is to know if we can have a phenotypic consequence to the transcriptional variability we have between plants. The second is when the differences in expression are happening during the life of the plant. So we are looking for that at one single gene because it's harder to do for the entire transcriptome. And then the third one is to try to grasp a bit what are the factors that might be involved in getting that high level of transcriptional variability. So obviously to do this kind of stuff, I've decided to focus on one gene to start with the idea to then expand and try to find if we can generalize that later. And to choose that gene, I had a lot of choice because before that, I did a transcriptome-wide analysis of variability, and I had hundreds of genes with high level of variability between plants, and then how to choose a good model to work with. And so basically, it was a bit of guess, luck, and in the end, I decided to focus on genes that are involved in nitrate mutation in plants. So the plants, they need nitrate to grow. They basically need carbon, nitrate, and a few other stuff. And that nitrate, they get it from the soil thanks to transporters. And here you have a list of main genes that are involved in nitrate mutation. You don't have to remember them. But the ones in red are the ones that I did find as highly valuable in expression between plants. And the proportion we have here is higher than the proportion you have normally in the genome. So clearly there is something happening there where we have a lot of genes that are highly valuable. And then if you put that in the context of the life of the plants, when plants are growing in the wild, the amount of nitrate you would have in the soil is hugely heterogeneous. So you would have areas with high level of nitrate and areas with low level of nitrates. This is at a bigger moment, but it is also changing with time. I mean, if you have a rainfall, obviously the amount of nitrate is going to change. If the plant is in the field and then you have fertilization by the farmer, the amount of nitrate is going to change. So if you think about that, then maybe that could become a good example to try to focus and find if there is some benefit behind that, which spoiler, you haven't found because we cannot keep plants by lack of nitrate. The model I'm using is NRT21, which is a transporter of nitrate. So it's a protein that transports nitrate from the soil inside the plant. And the expression of that gene is highly variable between plants. And what we are using is a reporter light. So we have the luciferase, which emits light, fused to the promoter of NRT21. And here you have the level of lights that are emitted by the luciferase in the plants. So in fact, here we have the food plants, but you only see the roots because that gene is only expressed in the roots. And you can already see by eye that we have quite big differences in the amount of luciferase signal between the plants. And to quantify it and not just say, oh, I see species. We use the coefficient of variation. We do that usually with around 30 plants per genotype per condition. So it might look really small for people who are working in bacterial analysis, but for us, it's already quite a lot of work, and it's the only thing we can manage to do in a reasonable amount of time when we want to start looking at multiple environments, multiple genotypes, because we have to analyze them in a 2-3 hours window and cannot do more than that. So technicalities. So to prove that this gene is indeed highly valuable, we decided to measure the coefficient of variation and to compare it with another reporter line that has the 35S promoter, which in plants is used as a constitutively expressed promoter. So the level of expression of that gene is very similar between cells and is very similar between plants. So it's a locally variable control, and you can see that compared to the 35S, we have a high level of variability for our gene of interest. So in our hands, like we have found in the RNA-seq, it is indeed highly variable. So now that we have that, we can try to answer these questions. And so the first thing we did was, okay, can we see a phenotypic link between link to this variability? So for that, just a bit on what this protein is doing. So I said it's a transporter. So NRT21 is here, and it's involved in transporting the nitrate from the soil to the plant when the concentration of nitrate is low. So we call it a high affinity transporter. And when the concentration of nitrate is high, it's not used at all by the plants, but it's other transporters that are used. And we can actually measure using labeled nitrate, the amount of nitrate that is coming inside the plant in an experiment, specifically for the high affinity transporter, where NRT21 is saying 80% of the high affinity transporter. So basically, we are measuring the activity of the NRT21 protein and how much nitrate is coming into the plants. That was never done on single plants, so we have done that on single plants. And we have done that for plants where, before measuring the influx of nitrate, so the activity of the protein, we also had the luciferase signal for that same plant. So we had the activity of the promoter. And then here, every point is a plant, and every color is a replicate. We do plenty of replicates. That's just a bit of what we have because we want to be sure of our results. That's the purpose of working on variability. And you see that for every replicate, we have a really good correlation between the two. So namely, when you have a higher expression for that gene, the protein function is also increased in the plant, and we have more nitrate coming inside the plant. And this was done for plants that were initially grown with low concentration of nitrate, where normally this transporter is the one mainly active. If we do the same, measuring still the activity for NRT21, but for plants that were initially growing with higher concentration of nitrate, where that protein is not used at all by the plants, then we completely use the correlation, which I find is quite a nice result. But still not a proof that there is a relation between the two. And for that, now what we need to do is to produce plants where we remove the variability for NRT21, and in plants that takes a year, so sorry, I don't have it yet, and see the impact of this. Okay, so it seems that we could have potentially different consequences. So everything I've shown you, there is high variability for plants we've measured when they are like something like 10 or 11 days old. It's like a quarter or a third of the life of plants, type of plants. But where are the differences happening if we start looking from the beginning of the life of the plants? And that's what we're doing. So I have to start the movie here. So still using luciferase imaging, we can do that for plants while they are growing in normal artificial lab conditions. And we measure the signal every 4 hours for our luciferase reporter. And you can see that for while the plants are growing, we have some plants with more or less signal happening. You can also see that we have a lot of plants where nothing is happening. And that's because these plants are in fact dying. We have them growing with low nitrate condition without sugar. It's really suboptimal for the plants. And we believe that these plants don't manage to make a key transition in the life of plants, where they move from the reserve of carbon that comes from the seed to the carbon coming from photosynthesis. So they cannot make a transition from the heterotrophic stage, where they get the food from the seed, to the autotrophic stage, where they get the carbon by themselves. And it's a good way for us to know when this transition is happening. So in a way, that was quite nice that we had that problem in our experiment. And so then we can draw the line of the signal for every plant. So you can see that we have a peak of expression at the beginning, and then we have day-night fluctuations for that gene, which are known. And you can see that for some plants, the signal is continuing to go up, while from others, it stagnates or even goes down. And if we continue for many days after that, we maintain these two groups. We sometimes have a few plants that will change group, increasing expression or reducing expression, but we have done that for more than 20 days, and we managed to keep these two groups all the time, with really good correlation that don't go down this time. So for you to look at these, just two plants in particular, so you can see with your own eyes and not just the graph. If you follow the purple and the blue line, so the blue one is where the signal is go down after some point, and the purple, it continues to go up. And that's more or less now that you will start seeing differences between them. At the beginning, you cannot see any difference between the two. They are behaving the same way, growing the same way, signal the same way. And so the purple line is when we see an increase in variability, and the gray line is when you have that key transition in the life of the plant. So you see that we have the differences in expression happening after that key moment of transition of the plant, a couple of days. Usually a couple of days is what we see in all our experiments. So now that we know that we have differences between plants established a few days after that key transition, and that it is not the case for the 35S, and that it is transmitted through multiple days, then the question is, are the differences in expression transmitted through generations? So for that, what we did was to take plants with high, medium, or low expression. We do a selfing, so they do self-fertilization, which is quite useful because then all the descendants from a single mother, they are all genetically identical. And we look at the signal we have. And so what you have here in the y-axis is the level of signal in the offsprings, and in the x-axis is the signal in parents. And we have absolutely no link between the two, so it's not related to the next generation. So it's like every generation, whatever the parents had as a level of expression, you again have the same spread of expression happening that is recreated a few days after that transition between heterotrophic and autotrophic state. So now that we have seen that, we go, okay, now we want to understand why this is happening, how it is happening, and we want to know the factors. And before going into doing mutants, because we had absolutely no idea of what might be these factors, okay, let's narrow down the possibilities. And to narrow down the possibilities, what we did was to do RNA-seq on single plants, so 24 RNA-seq on single plants, and then look for genes for which the expression is changing in the same way as NRT21 in the different plants. The idea is that if the expression for these genes change the same way as NRT21, then maybe they are in a way involved in pathway that might lead to that variability in expression. And so that's what you have here. We have done a hierarchical clustering where every column is one plant, every line is one row, and you have the color code for expression level. And the NRT21 gene is in this cluster with other genes that behave similarly. It's not easy to see in there, so I've been extracting the actual data from that cluster, and you can see that indeed NRT21 in black has an expression in different plants that is similar to the other genes in this cluster. So now we can look at the functions that are associated with this cluster by doing gene-ontology enrichment, even if it's not perfect, it gives us ideas. And so here are the functions that we get. So we get things related to development, things related to photosynthesis, but nothing at all related to nitrate nutrition. And I had a look specifically at all the genes that are involved in nitrate nutrition, and all of the transcription factors that are known to regulate that gene, and none of them are co-expressed with NR2. So clearly, by doing this analysis, we are finding new ways of regulating a gene that has been studied for like 20 years, on which we know more than 10 transcription factors, and still it shows we are missing a lot of information that we now find thanks to this variability. So I'm going to skip that because it's a bit complicated, and I think I'm running out of time. But here is a summary of what we think might be happening, and it's a hypothesis that we want to explore in the future years. So during this life of the plant, so at the beginning, the plants, they are in this heterotrophic state, so they get the carbon from the seed. And then you have that transition, and then they become autotrophic, so they grow thanks to the carbon that they get from the photosynthesis by themselves. And the idea that during that transition, a lot of things is happening. You have a lot of reconfiguration of the chromatin, of the transcriptional networks, and so on. So quite a lot is happening there, and a few people are working on that, but not at a single plant level, obviously. And so what we're suspecting is that at this moment, the plants go out of that transition with different metabolic states. So we have some plants which have a low metabolic state where they do low photosynthesis, and in the plant you need to have similar amount of carbon and nitrate because both are essential to make amino acids and proteins. And so when you have low carbon, you also have low nitrate. And then you have other plants with a higher metabolic state, with higher level of carbon and higher level of nitrate. And that this state is just propagated through the life of the plants. And then what we suspect is that after you have that difference in metabolic state is when you start to see differences in the expression for our gene that are appearing. So for us, what we're looking at is not necessarily stochasticity at NR2 itself, but we are looking at variability that comes from the stochasticity that is somewhere else, which we still have to find. And so with this, I want to thank all the people who've been involved. So the CRM team, and in particular, Alexandre and Charlotte Lecuyer. So Charlotte is here, and she has a poster she presented yesterday. She's looking more at the impact of chromatin on that, which I have not talked about on purpose. She has all the things to show about chromatin, and she has great results. And the collaborators have been really helpful for data analysis and creating these nice movies. And thank you for your attention.
I really like this last result because normally I think of the origin of the stochasticity or noise or variability in gene expression and then something happens after. So I really like this idea that the stochasticity is something else that is kind of causing this variability, right? So it's your feeling that this would be the major driver of gene expression variability is stochasticity, something else that might be exposed to what you said metabolic state.
I think that really depends on the genes we are looking at. So, for example, genes that are really involved in the response to stress, I think that would be a very different story. But for genes that are involved in metabolism, that might be something that we've discussed before about size of the cells or metabolic state of the cells and stuff like that. I think probably here we have something similar. And one thing I'm starting to suspect is the quality of the seed that comes from the model. So we know that there is a variability in the quality of the seed, and so we're trying to think of ways to test that maybe some seeds will have better quality than others. And so if you have a low quality seed, then obviously that transition is a bit more complicated than if you have a high quality seed. You go out of that transition in different states, and then this is propagated through the life of the plant. And obviously this is just one gene, And for me to try to go further and to prove that, the idea would be to do single plant variability before, just after the transition, and at the later point, to really have the temporality of what is happening. That's just one gene.
In a follow-up on this last point, how much can you control for the quality and state of the seed?
That's a really good question. So we know that at least for that plant, Ariadne so the model plant that everyone is using. So we know that the first seeds that are generated are not really good, and the last seeds that are generated are not really good, and the ones in the middle are best kept in terms of ways of creation of the seeds. So what we are just doing now is to harvest the seeds at different moments. And to try to see if then we see differences in the expression of NRT2 in these different batches of seeds related. So we've done it once, and it seems to go in that direction so that we have a higher expression for NRT2 for the middle batch and lower for the two others. But I want to replicate fully the experiment, and obviously every time it's like 3 months of growing the plant and waiting the seeds and so on.
So this is maternal provisioning, or?
So, when I say a carbon from the seed, that's carbon that is in the first, that are not true leaves. So the first leaves that come out are not true leaves, and you have most of the carbon there in the form of like lipids and stuff like that, which are degraded to get the carbon at the beginning of the life of the plant before you have photosynthesis. And so how much you get there, I'm not sure exactly what is controlling it. There is you have a few things on literature about that, but not that much. And I've not found a way to get the amount of carbon you have on the seed without destroying it, so that you could measure it on a single seed and then grow the plant and then have the rest. So, yeah.
Thank you.
Thank you. Do you think there might be a feedback between developmental stochasticity and expression stochasticity? I don't know if the leaves happen to develop well and go for one straight.
Yeah, that's a possibility. So in general, we see a nice correlation between the expression of that gene and some phenotypes other than the one I showed, like the actual growth of the plant. So when you have a higher level of expression for that gene, the plants are bigger. And then possibly, because they are bigger, they also have a higher metabolism. And so it's like, just maintaining the system. But we have another hypothesis with Charlotte that maybe it is something about chromatin, where really you have different levels of chromatin in the plants that are established at that moment that are maintained over time. And I'm not sure about the two, or maybe it's the mix of the two. Life is complex.
Yeah, just as a natural consideration offer that everything like floral phenotypes or flowering type, for example, is there a connection between foreign phenotype and the expression of this gene?
Yeah. No, I've not looked at adult phenotypes, except that looking at the next generation, just because we are growing them in petri dishes and not on soil, because we want to see what is happening in the roots, which you cannot do on soil. But that's a really good idea. Yeah, one thing we try to do is to put them in the context where we remove all nitrate and see if we have a link between the level of expression of that gene and how they survive to nitrate starvation. But then we maintain them like that for more than 20 days without nitrate, and we didn't manage to kill any plant. So we could not make a test, saying, okay, that one has more nitrate and it survives better, because we cannot kill the plants. So I'm trying to think of other ways. Yeah.
Thank you very much.